here. And I'm going to share my screen. All right, so you should hopefully see my screen, and then if you want to make any comments, I will try to be watching out for that as well.
I can see it.
So looking at our newest lab chapters, chapters 28 and 29, we get to look at our animals, which is really fun. And so first thing to understand about animals is how do you define the different parts as you're looking through the specimen. We want to be able to look at the different regions. And so that's where we start here, the anatomical orientation, looking at 3 body planes. That you have 3 body planes here color-coded for you. And so these 3 planes, you have the first one being the medial or sagittal plane, which is in red. And so that one's dividing it from your left side and your right side. Then you have the transverse plane, which is in blue here. And so that one's going across, like a cross cut of the body. So you can think like a tree stem. As we looked at tree stems, we cut it across and got to see all the rings. And so that is your transverse plane. Then you have your frontal and coronal, which is front and back separation from the front side and the back side. So let me check here. Still getting used to Zoom. Sorry about the delay. So make sure I'm not missing anything here. Let's see, where's our chat? Here we are. Okay. Make sure I have that up too. Okay, and so looking at another example, which is more closely to what we would see in that it's in fish. So you have the anterior end by the head, the posterior end is the tail, dorsal side is the top side here, which is usually darker in coloration. So, you know, D for darker, and the ventral is lighter, and it's on the bottom side. And then it's harder to see in this picture, the lateral sides, the right and the left side. And so in this chapter, we look at a wide variety of vertebrates, invertebrates, just invertebrates, and we will see anywhere from sponges to earthworms. And so here in the Phylum Porifera, which is sponges, you have different components of sponges that we'll look at in terms of, by picture here, the first one being spicules are crystal rods that help for protection and support. And so those are right here. And so those will be in part of the center of the sponge, as well as like on towards the outside of the sponge for protection. And then looking at the next one here, the osculum is where water will leave. So osculum is any like opening that water will exit. Then you have the ostium, which is where water enters. So there's a very distinct direction flow of water because the choanocytes are going to be filtering that water. And so, let's see here, no, yes. So yes, it can be anywhere on the sponge. So here's just like a diagram of that flow. And so it varies from sponge to sponge where the water will come in and leave. And so this general one is the simplest sponge. And so there's ostia all over, and then usually the osculum will be at the top. And so usually there's just one osculum per sponge. And so water can flow in anywhere and usually it has one exit. And then for the central cavity is the spongocoel. That's the center of the sponge right here. And let's see, radial canals are something we'll see on the next slide better. So there are three main body types of sponges. The first one being the asconoid sponge, which is shown here. And so water will enter through all the ostia on the sides. So basically on the sides of the sponge, all here and here. And then it will go, it's the simplest type, so it just goes straight through that opening. And then the choanocytes are on the inner lining of the sponge, which is shown here in this darker color. It lines the whole center of the sponge, and then the spongocoel. So they filter the water, and then it gets pushed out to here, and then it will then go through the top opening of the osculum. And so that is the asconoid sponge. There's no, there's no specialized portion. It's just in through the ostia, choanocytes filter it, and then it leaves through the osculum. And so in appearance, it looks like a smooth vase, which you see in that picture there. And that brings us to the second type of sponge, the syconoid sponge. This is a little bit more complex in that the ostia will open up into canals called radial canals, which is shown here. Radial canals are like, kind of look like finger-ish projections from this, side of the sponge. And so there in the radial canals where they will have choanocytes that will filter the water for food. And then from there, that water goes out of the radial canal into the spongocoel and then out through the osculum. And so this looks like a rough or rigid vase in appearance. That's the second type of sponge. Then the third type of sponge is the leuconoid sponge. And this is much more asymmetrical and disorganized. And so here you can see it flows in through ostia again, and it goes to these chambers, these little cavities where choanocytes live. They'll filter for that food, and then it will go out to the spongocoel, which is usually a cavity in its center, and then out through the osculum. And so it's a much more asymmetrical and disorganized, but it has distinct regions where it flows in, which is, along the whole body of the sponge, and then usually one main osculum that flows outward. And so you can see that here it's much more asymmetrical. And so that's the three types of sponges. And so in lab, we have some specimen that we look at. And so the first one is a Grantia sponge, which is also referred to Scypha. And it is a syconoid sponge. So it's going to have radial canals, what you see here. And so they cut the sponge in half, like from the top to the bottom. And so you can see, just like our diagram in the first thing here, you can see the same kind of imagery, but with radial canals. And so here you can see openings where water can flow in. Let me change my color. So openings where water can flow in are ostia. And then they have the radial canal, which you can see here, opens to the spongocoel. And so that's what you're looking for, that opening there. And so here, all the little darkish red-pink are choanocytes all along there and here. And so then looking at it closer, this is from our actual lab picture one that you can see the opening of ostia, radial canal here, if you're looking at water flow, it would be the opposite direction. And then the spicules, spiny, either silica or calcium carbonate rods. They come in different forms, but in the Grantia, they come in a 3-pronged spicule, and so it kind of looks like a Mercedes logo, which is what I like to tell the students. And then here's a whole specimen of that same species, Grantia. I think this one's dyed green. It's not normally green, but you can see this is the osculum here, and then osteo would be all along the sides.
So is it usually clear?
And then just some specimen that we would have in the lab. So here are just 4 different ones. This is the one that we're looking at as Grantia. That is pretty small compared to the other ones. And then you have some more specimen, like this one looks like a rough vase. That's probably parasitonoid. This one as well. That one's probably leuconoid. And then that one's probably leuconoid too. I don't see any asconoid there, from what I could tell. So that brings us to the cnidarians. So the jellyfish, the corals, Hydra, they're in this group. And so they get their name from their cnidoblast stinging cells. And so they have 2 body types, medusa and polyp, which are shown here. And then you have just some examples from lab. So we have a sea anemone, jellyfish, coral, hard and soft coral, and then more coral here. And so for this section, I like to focus on a couple examples, being from the hydrozoans, where the polyp form is dominant. We have 2, the Hydra, which is polyp form only, and then the Aurelia, which it has a little bit both, and then Scyphozoa, which is medusa dominant form, which we have the Aurelia, the moon jellies. And so let's take a look at those. So the hydras are shown here. Hydras are polyp form, so they're going to attach to substrate at the base. They have tentacles that are surrounding their mouth for feeding, and they're capable of both asexual and sexual reproduction, which are shown here by budding is the asexual, and then the sexual has the gametes, the ovary and spermaries here. And so looking at our lab, we have some closer images of those here. And so this one is not budding, or you don't see the reproductive gametes, so that's probably a youth Hydra. And so looking at the next specimen is the Aurelia. It's much different. It looks more like a plant. And so here you can see for Aurelia, we have let's see, a couple different, a couple different parts of it. So you have first a polyp stage, which is stationary, and then different buds. And so these buds coming off of the stalk, one forms medusas, which is right here, and you can see it's you can see the bell top on the topmost portion there, and then you have the hydranth, which is right here, and that's for feeding. And so hydranths are for feeding, and the reproduction ones are medusas. And so we have a specimen there. You can look and see which ones are hydranths. Most of these here, the ones forming medusas are a little bit less apparent there. And then for the Aurelias, they're from the Scyphozoa, so they're predominantly Medusa. And so those you can see have different stages in this life cycle where they do have a polyp stage, and then from there they will bud off and form the adult Medusa. And so for our lab, this is what we have for them. They also have a slide, but it's usually hard to actually understand. But you can kind of see the ends of the Medusa bell, and then the center has these four like C's on them. And so that is how you can find the moon jelly really easily. That brings us to the flatworms. So phylum Platyhelminthes, you have four classes: the free-living Planarians, parasitic tapeworms, parasitic flukes, and then ectoparasitic flatworms. So let's take a look at those. The first one, the free-living Planarians, we have Dugesia is our Planarian that you can tell it looks very similar to what we saw in lecture, with the gastrovascular cavity is very apparent under a microscope, the pharynx region, along with the eye spots and auricles. Those are all key indicators of the Planarian. And so here you can see that there are auricles. The auricles are the little projections from the side of the head. They're used for tactoreception and chemoreception. And then the eye spots are kind of giveaways for this as well. So we'll compare those to other ones that are parasitic, will not usually have those structures. And so here are some images from lab. Again, you can see the projections, the eye spots. Those are kind of key indicators. Onto the second group, the parasitic tapeworms, which is shown here, class Cestoda. So here you can see the tapeworm body consists of the scolex head region and then the tail region, which are made up of repeating segments called proglottid, which is there. And so we're looking at the tapeworm. As you move down the units, they start off becoming mature to eventually form eggs. And then after they form those eggs, they get fertilized, and that will now make it a ripe or gravid proglottid. And so that's ready for dispersal. It has basically offspring that are ready. And so that looks more, I guess, bush-like in terms of netting there in the dark center. Those are all the eggs. Whereas here you have distinct regions for ovary and testes that make the gametes. And so that's shown here, that the mature proglottid is usually not as dark in appearance. Everything looks more one color. You can kind of see some distinct formations of different reproduction organs. But in the ripe proglottid, which you can see in this corner, there we go. You can see in this corner, is much darker and netty, more bush-like or tree-like in appearance. So that's your ripe proglottid. It contains eggs. And then looking at the scolex, you can see the suckers pretty well. Hooks are kind of hard to distinguish there. And then moving to flukes. So flukes would be the third group of class Trematoda. Here's your Chinese liver fluke. That's known for parasitizing dogs, cats, pigs, and humans, where they have most notably two suckers, one with the mouth, which makes is oral sucker, and then one on the bottom, the ventral sucker, and that's to attach to its host and then to take in nutrients. And so that's how you can know this is parasitic and that it needs one to attach and one to eat. And so here you can also see for the Chinese liver fluke or just liver fluke, they also have these two long branches here. Or cecum or ceca, those will be part of the digestive system. So that's a key indicator of which fluke it is. You're looking for that ceca, and that's your Chinese liver fluke. And so as you can see, there are more structures, but I like to focus on how you tell these things apart. And then looking at Trematoda class with a sheep liver fluke. This is one of the largest flukes, so under the microscope you only see part of it because it's so large. And they live in liver and bile duct of cattle, sheep, and humans. And it looks much more different, but still it has the two suckers, so one here anterior and one ventral. And so flukes are known for having those two suckers, one for attachment, one for feeding. And so the rest here you can see it has yolk glands along there, intestines here. So it's much more different in shape. In the front shape, this shape can distinguish it from the liver fluke as well. So then looking at blood flukes will be the last in this fluke group. They're found mostly in Africa through contaminated water, and they will stay what they call in copula, where the male and the female will stay together so they can reproduce and produce more of them more efficiently. And so here you can see both male and female together. And so they look more like a typical worm. And so that's how you kind of distinguish the three flukes from each other. And so that's what you see here. Your liver fluke, your Chinese liver fluke, your sheep liver fluke. This is a picture from our lab that you can see the oral sucker and then the ventral sucker, and you can't see anything else really, it's so large, and then the blood fluke. That brings us to rotifers. So rotifers are very, very tiny organisms that were once thought to be protists, but now we know they are not, as they have a complete digestive system and are capable of many other things that protists do not have. And so here you can see this one's labeled, and so part of it kind of is hard to identify with all the labels. And so here's another example of that, a rotifer. But I like to stick to a labeled one. But they have crowns, cilia, the crown of cilia, like a circle of hair-like projections on their head. They have a transparent body. Like I said, had complete digestive system. Interesting facts about these are they undergo cryptobiosis, which is a state of dormancy where they basically stop functioning. And then can resume functioning another period of time. Another thing is that some of these species are females only, and they're capable of reproducing with unfertilized eggs. And so here I can see the body of it, just like the other one. And then the cilia are somewhere up here, and then some is at the head. And then lastly, earthworm. So in lab, we normally dissect earthworms. So I like to start out with looking at a slide for earthworm. And this is a cross-cut. So this is where to go back here. And remember, this is your transverse cut in blue. Transverse cut of the earthworm. So you can see its skin, and then the longitudinal muscle are these hair-like projections going inside. So that's the muscle that helps it move. And then the center of like a crescent moon, that's the intestine. And so the center of the intestine, this is right here. This is where all the soil would be. And then looking at this one, you can see that there's the dorsal is labeled. So this is the top region, and then this is the bottom. But if you weren't sure, you would look for the nerve cord, which is always on the bottom of the earthworm, and that's right here. And so that's what you'll want to be able to do here. See that this up here is the dorsal blood vessel. This is the ventral blood vessel. I can still missed it. There you go. Ventral blood vessel, and then that's because this right here is the nerve cord. And so you can tell muscle, and then the intestines is the center of the intestines right there. Yes, so the nerve cord is always going to be on the bottom, the ventral side of the earthworm. And so if you weren't sure, you just look for that. If it's not green, if it is it clear. Oh, so the intestine, so this right here, I was just drawing in green, but it's, in this case, this earthworm intestines is clear because there's no food in it. But if it had food in it, it'd probably be dark and it would look like particles in the food. So I hope that helps clarify it. Are there any questions you have about this chapter? Okay, well, thank you for joining me today, and let me know if you have further questions, but you'll have an assignment over this section, so hopefully you're better prepared. I'm going to pull up Zoom now. Let's see. And I'll see you later.